So, thank you everyone for being here today. It's a real pleasure to have been invited. So my name is Philippe Wilson. I'm a professor at Nottingham Trent University in the Medical Technologies Innovation Facility. And the work that we do in my group is around human and animal medicine. And we do both human and animal medicine because lots of things that we can learn about the human, we can translate into the animal and vice versa. So where have I come from? Tullis's favourite, the Shropshire Star. So my beginnings were in farming. So I grew up on a rare breed farm, farming Dexter cattle, Hebridean and Soay sheep, and lots of chickens. I'll press the wrong button. Press the same button again. Maybe not. No, I've got it now. It's fine. Let's just get Tullis stood up again. We'll keep doing it again and again. So and then I ended up becoming a poultry judge, which was great fun while I was at university. So I did my university degree in Bath in chemistry. I spent a bit of time in the veterinary school at Bristol as well, which actually helped to shape how we ended up doing research. And I sit on the Comparative Medicine Council of the Royal Society of Medicine. What does that mean? Is that recognises the fact that you need both human medics and animal medics in order to be able to learn a lot more about the health, the welfare, diseases across the world. We learned that a lot during COVID, right? So pangolins being a bit sick, ending up killing a lot of people. That was pretty much the story of COVID. And we didn't really recognise that properly until the pandemic happened that forced humankind to realise that there were these translations between diseases between different animals. And I spent a year as scientific director of the Rare Breed Survival Trust in 2019. And that was pretty much trying to get the charity onto a scientific keel, which I think I did a little bit. So we published a lot of papers, did a lot of work with my good friend Andy at the back. And I think we did a lot of good in the RBST. So where do I live? Well, this obviously isn't my house. But when I'm at work, which is where I live most of the time, this is MTIF, so the Medical Technology Innovation Facility at Nottingham Trent University. And what it does, it's 4 walls with lots of equipment in it. So what we do is we have businesses, we have companies like Stallion AI, we have charities come and see us with problems, and we answer them. Problems in human medicine, animal medicine, device development. And we're a strategic partnership with lots of universities, the NHS, and also various charities that do all sorts of good things, particularly in cancer and human and animal disease. And we've got 2 facilities, one on our Clifton campus and our Boots Enterprise Zone. And here we can manufacture drugs as well and do all sorts of wonderful things like that. So really the pipeline of health from one end to the other. And what do? We've got an MRI scanner, so we can scan humans and animals, find out lots of stuff about their cells and imaging. We do drug testing, we've got microscopy, we develop medical textiles. You might have seen on the news this morning, as soon as I woke up, BBC News was talking about a new foot sock thing that some of my colleagues at NTU have developed, which is for elderly people. They can wear it, it's got a sensor in it. It measures how they hobble around a little bit. Then if something that happens, if they fall over, for example, unusual movement, it alerts the hospital. So if people fall over, they haven't got access to their phone. It does that. This is the sort of fun stuff that we do at NTU. So we also have clean rooms, we do cell therapy, and we also do wonderful things like metabolic profiling and genomics I will talk about today. And we have an integrated theatre suite. So this is a great piece of kit. It's effectively an NHS theatre or a veterinary theatre. But if people have new endoscopes or technologies that they want to incorporate into operations, they can actually come in and use it in our integrated theatre suite, test it out, rather than hiring out an operating theatre in a veterinary hospital or an NHS hospital, having to cancel all of the cases for that day. They can just come and do it here where we have no cases. It's just for them to test stuff out. So this was a really great idea that some of my colleagues had when we were developing this. So what am I going to talk about today? And I promise I won't spend too long talking because we all want to move around a little bit after today. But we're going to talk about omics. And omics is this wealth of data that we can get from human things, human and animals. So we can get information from the genome. What is our base code that tells our bodies what to do? And that's the same. We have genomic code in everything that's living. Our epigenome, how does our genome change with the environment? We know that if people are exposed to radiation, their genome can change and they can get things like cancer and stuff like that. Transcriptome, what does that genome sort of tell the body to produce in terms of proteins and different molecules? And then the metabolome, so when you go for a blood test, when you go for a urine test, what that's testing is the metabolome. And that combination of what we call metabolites, individual chemicals, that sit in your urine, saliva, or blood. And that each of these different omes tell us a lot about the living thing that we're looking at. And they can tell us whether they're healthy, and they can tell us whether they're diseased. So whether this is going to work or not with my excellence at technology. Oh, amazing. So genomic medicine, this is a new era that we are in, is that we are now able to sequence, to understand a lot more about genomes, more than we've ever been able to before. So in the canine genome, there are 2.47 billion parts, 2.47 billion letters. That's about 600 J.K. Rowling books, I think I read earlier. Not that anyone would necessarily want to read a J.K. Rowling book here. And 0.2% of that, that's not a lot, is different between people, different between dogs, different between individuals. And it's that 0.2% that actually dictates whether someone might get breast cancer, or a dog might get hip dysplasia, or something like that. So it's that 0.2% that we can detect, and then make decisions on how we treat, how we prevent disease, how we identify whether actually it's worth breeding with that individual. And we can only do that through techniques that are called sequencing. It would be nice for it to end just when I said the word sequencing, but never mind. And we at NTU have partnered with... Alice, can you calm her down? That's very exciting. I mean, I get very excited about Oxford Nanopore technology, I'm not going to lie. What Oxford Nanopore does, what sequencing does, it effectively reads, like a book, a whole book. And the technology is based on all these little holes, which are called nanopores, hence Oxford Nanopore. And through these holes, you get DNA that passes through it. So there's one of your nanopores, DNA, RNA, whatever genetic material. It gets tethered by a tiny, tiny little rope, and then it gets pushed through. And as the DNA passes through, it's red. That pore reads the DNA. Through that pore is an electrical current. And as the DNA passes through, whether it's one of those 4 Lego bricks that Andy was talking about, A, T, C, or G, if it's A, one electrical current will pass through. If it's T, it's a different one, and so on and so on. So actually, you're reading 400 bases per second of DNA as it goes through this nanopore, which means that we can actually sequence the whole canine genome, for example, in 8 hours. That's nothing. It took the Human Genome Project 30 years to get to the human genome. And now and it's only thanks to some of the innovations during COVID, which meant we were sequencing COVID genomes, and we were part of that effort at NTU, that we were able to get to technologies now that we can really put into precision medicine for animals and humans. It might let me change the slide now. Right, and so as part of sequencing, what can we do? Well, we can actually look at how related individual dog breeds are, or individual animals, humans, whatever. And this was about 2,000 individual dogs that were sequenced in the States about 20 years ago. In 2020. 20 years ago, it would have taken them a long time to sequence. And you can, I mean, you probably can't read that, but I'll make the presentation available to everyone. If we can do that, maybe we can't. I'll just say we can. It's extra work to tell us, so I'm happy with that. But you can see how each of these breeds is actually not necessarily related to the ones that are next door to them, but related in the grand scheme. And this relates back to what Andy was saying. If we go back in our ancestry to Battle of Hastings, we're probably all related in one way or another. So why would we bother sequencing? I mean, I think it's great, but let's put some pragmatism on that. So the Kennel Club in 2018, I think, started this Give a Dog a Genome project. And as part of that, they looked to find genetic mutations and develop some tests. And one of the tests was in the Shetland sheepdog. And there was a particular recessive mutation that caused this blinding condition. And they compared Shetland sheepdog genomes to those of other breeds that didn't have this mutation. And they found that there was one particular retinal gene responsible for what happened in the eye that led to this mutation. And they developed a DNA test for it in 2020. So sequencing can lead directly to testing for some of these really, really difficult conditions. So I talked about health in the last slide. We can also think about wellness. So what do I mean by wellness? I mean, okay, not necessarily we're treating diseases, but we're actually looking at making animals a bit healthier. And one of these examples is, for example, the microbiome. So a lot of you will have heard about taking probiotics, how wonderful that is for you and all that sort of thing. That works as well for animals, as humans, and all sorts of living things. We all have this microbiome. This is the bacteria, viruses, fungi that live in our gut that help us to be healthy. And there was a study done during COVID that showed that in humans, a healthy microbiome reduces depression, reduces anxiety, reduces conditions that you wouldn't actually link to necessarily physical health, but it was the microbiome, that combination of good bacteria and things that we've got that contribute to our mental health. Also, another study on toxoplasmosis in cats. So people who caught toxoplasmosis from their cats, there was a very, very clear correlation between those who had toxoplasmosis that you can get through immunological markers and the incidence of psychoactive disorders afterwards. And those things just hadn't been linked. All of these microbiome effects hadn't been linked before. And it works just as well for our friends, our dogs and cats and stuff. If they have a healthy microbiome, they will be healthier in themselves. There's been evidence, there's a couple of clinical trials going on at the moment in terms of how a healthy microbiome, taking probiotics, the right kind of probiotics, can help to support cancer treatment, can help to avoid all sorts of diseases as well. So it's all very, very intrinsically linked. And what you can do is you can take samples, and we do this on a regular basis, particularly with equines and felines, is we take samples from the gut and we can sequence them, like I showed you before, and get all of the different things that are in there, from fungi, bacteria, viruses, and figure out what's there and what should be there. So you can design probiotics to supplement what you've not got or what the dog hasn't got. So why do we want to do sequencing? Well, we can sequence to get rare diseases, so we can find out why one individual in an animal population has got all of these horrible conditions. But also, like Andy said, about Mendelian diseases. So how are diseases passed down through individuals through a whole population? And really the next step to sequencing, many of you will have heard of gene editing, I'm sure. It was what I sit on one of the government committees, UKGLE, which is UK Genetics of Livestock and Equines, very catchy name, as you can imagine they came up with. And that's the DEFRA committee for genetics of things that are like livestock. So that's why we've got equines, camelids, and all sorts of things like that in there. And one of the things that came out of the consultation when Brexit was taking place was, and I won't voice my opinion on Brexit because I won't voice it, but one of the benefits of leaving the EU, apparently, was that we were able to take advantage of gene editing. So the EU had a regulation that said that gene editing was the same as genetically modifying foods. It's really not. Scientifically, it's absolutely not. They're black and white, okay? Gene editing is effectively, you're not putting genes from different things in. All you're doing is you're correcting errors. So, if you were to misspell a word, what would you do? Why can't we do that with our genome? All we're doing is, Tullis may have genes that, actually I'll put you as a bad one. Tullis will have genes, may have genes that are not correct. They might have genes that have been toned down, for example. I might have genes that have been turned on. And all we're doing, I'm still a human, Tullis is still a human, we're giving Tullis some of the genetic advance that I might have. And so on. There might be lots of people who have far better genomes than I do. But all we're doing is we're taking natural changes in genomes and affording other individuals in that species the same natural benefit that those others have had. So it's not putting anything unusual like plant DNA or cactus DNA into people. I saw an article the other day, someone was claiming, although they'd probably be less spiky. Thank you. All it's doing is it's snipping. You're snipping a bit of DNA that's naughty, and you're just swapping out. It's like taking out that misspelt word and spelling it correctly. As simple as that. I say it's simple as that. It's actually quite complicated in the lab, but theoretically, it's as simple as that. So one of the articles this was 2018, so actually quite a while ago now, 5 years ago, they were already doing gene editing. Now it's a much more commoditized and available technology. Nobel Prize was won, I think, 2021 by Jennifer Doudna and colleagues in CRISPR-Cas9, which is the technology that you use to gene edit now. But there's an opportunity to treat muscular diseases in beagles. And then that was used to learn how to treat muscular dystrophy in humans. So the dog benefited and the humans benefited. So there's real potential in these technologies that are just now starting to be used. So you may have seen in the news, I think it was about January, that there was a treatment, the first treatment for gene editing was authorized in a young child in the NHS. Pretty expensive, but it took that child from probably dying within a year to having a life. And it was because they'd inherited a condition that meant that a gene had been turned on. So you just turn that gene off and carry on. And gene editing, like any other technology, will keep getting better and better. And the list goes on with what we can do. You could support the heart health of dogs that suffer from heat stroke. You could look at extending lives of terminally ill dogs through advanced cancer therapies. We know in humans, for example, there's a gene called BRCA2 that means that women are more at risk of getting breast cancer. They won't necessarily get it, it makes you more at risk of getting it. But if you could turn off the BRCA2 gene, which you can, then that would prevent a lot of breast cancer cases. And it's the same with a lot of cancers, a lot of endemic conditions. So going on from gene editing, we're talking about changing the misspelling. What's the next step of changing the misspelling? Why don't we just cross out the word and print it out? So this is what we use for biobanking at the moment. Look at Tullis downstairs. He's got all sorts of wonderful big tanks that have got millions of samples in them. Tullis actually will probably use all of those millions of samples. But when I was at RBST, for example, we were biobanking, I don't know how many samples a year, most of which were never used. Paying for tanks to store all of these samples, all of this semen, rarely ever used all of it. I mean, none of it was all used, right? What if we could use sequencing to read genomes, so we knew what the letters were, save that in a Word document, not in a freezer, save it in a Word document. When we need that genome, we just print it out. That's the next step, and we're doing that now. We're able to print DNA when we need it. And that's the next step from sequencing through to DNA printing. And this is me being a bit facetious and printing a heart. We can't do that yet, but that's the next step. So once we can print DNA, we can put that DNA into cells. Those cells can proliferate. What you've done is create synthetic life. So this is the next step of being able to treat disease where people lose limbs, lose organs. Same with animals. So going back to our omics spectrum, we've talked about the genome and how wonderful and fun we can do new things like print DNA. At the other end of the scale, we've got the metabolome. So this is an analogy that I stole from one of my colleagues, and I won't reference him so that he doesn't get the credit for it. Genomics is effectively a huge piece orchestra that is ready to play. That's your genome. When you're born, you're given your genome, your orchestra. Okay? Further down the line, metabolomics is that orchestra playing a symphony. And it could play a symphony one day. You could give a blood sample one day and you're fine. Could play a different symphony the next day. You give a blood sample and, oh dear, you're lacking folic acid. So all sorts of different opportunities. So this is instantaneous measurement at a point in time. Genomics is measurement, what you're given. What's your playbook from birth? And metabolomics is basically the study of all the molecules that are in your blood, urine, saliva, or tissue. You can also look at tissue. And they measure your health. And some of these small molecules are sugars, fats, hormones that you can look at. And, you know, everyone can understand if something happens in your environment, you eat something, obviously your salivary metabolome is going to change. If you eat something spicy, you're going to have some spicy molecules that are in your saliva. And the same thing, and that's an environmental change, the same thing can be said if you've got a disease. And the same is for a dog, same is for cats, same is for any living thing. Whether you eat something or you've got disease, that's an environmental change that means that you get a change in your metabolome. And that can be measured. And it's measuring the metabolome that gives us insights into different kinds of diseases. So we did a lot with what we call NMR metabolic profiling, very catchy as well. I came up with it, thank you. So what we use are miniature MRI scanners. That's that we came up with. And we look at any biofluid, any fluid that comes from a living thing, and we can look at all the molecules that are in that fluid. And from looking at all the molecules, we can say, oh, right, okay, you've got upregulated something, you've got lots of this in your biofluid. That probably means that this is wrong. We can go back to the genome. We did a really great piece of work with some of my colleagues from Linnaeus and Mars Veterinary Health. So I do a lot of work with them. I sit on their Veterinary Research Council. Many of you may know them because they own most of the veterinary referral hospitals in the UK. We were looking at metabolic profiling of chronic kidney disease and cats that have been treated for hypothyroidism. And we could detect that much, much earlier than current tests. Because normally what happens is that you measure how fast the kidneys are filtering the urine in a cat or in a human for chronic kidney disease. And we could find markers of that much, much earlier than you could through current methods. And it's the same through, you can look at obesity. You can detect whether an animal, a human, is actually going to be at risk of not being able to process foods the same as another. There are lots and lots of exciting things you can do. And this is where the idea that dogs can smell diseases come from. Because what does our sweat do? All of our sweat is saliva. It contains lots of molecules. So where you've got people with Parkinson's, this was a huge thing recently. People with Parkinson's, they had dogs that could smell Parkinson's because there were molecules coming out in the sweat that the dogs could detect, which is bonkers, but true. And it's exactly the same principle as our metabolic profiling. All a dog that's sniffing some sweat from someone with Parkinson's is doing is metabolically profiling that. But we can also do that for the dogs. We don't need to use the dogs as our sensors. And where does the real power come in? This is the trick, is where we put things together. So during COVID, everyone will be thrilled to see another lateral flow test. But there was a really good thing that happened to lateral flow tests, is that they became a democratized technology. Everyone knows in this room how to do a lateral flow test, right? If before the pandemic someone had given you that, can anyone say they would have? I don't know. I wouldn't have known how to do it, and I'm supposed to be a scientist. So it's now a technology that people know how to use, and we need to take advantage of that because people, pet owners, horse owners, can be able to use these technologies to detect lots of different things. So all a lateral flow test is it's effectively a combination of the technologies I've shown you before, but on a slip of paper. So what it was doing before, for us during COVID, we were just looking at COVID, but lateral flow tests can do a hell of a lot more. So at the moment, what we're doing in my lab, one of the things we're doing is developing lateral flow tests to detect brain cancers. We can do that because there are molecules that are upregulated in brain tumours that aren't upregulated in people who are healthy. And for anything that has a different amount of molecules for unhealthy to healthy, you can do a lateral flow test for. So really, the future is us being able to use lateral flow tests to do really exciting things like detect diseases early, look at genomes. We can even look at the genome, some parts of the genome with lateral flow test. So that's really important. We can look at all of these conditions. I just took this as an infographic from Google for the top 5 apparently painful conditions for dogs. All of them we can do with lateral flow test. So take those, any other condition, most conditions we'll be able to look at. And so the real power is when we combine this sequencing, like I said, we can do gene editing. We detect the problem first. The sequencing informs that. We gene edit it, solve it. We've got all the technology to do that now, and we're right at the start of it. So it's a really exciting time to be in the health space. So what have we talked about in a very quick time frame? Huge potential that we've got to combine these technologies in human and animal health. And the UK, because we've got our own gene editing legislation now, we can really plough ahead and do some really exciting stuff in terms of developing new technologies, but also consolidating that and learning more about sequencing. And I talked about Oxford Nanopore, great UK company that really grew during COVID and is now, you can take some of these, you take people using these flow cells, so they're about this big, and take them into the Amazon, connecting them to their iPhone and sequencing the genomes of new plant species they found in the Amazon. That's how sequencing has democratized itself. And 20 years ago, a room this big would have one sequencing instrument in it. So we can really take advantage of learn stuff from the genome